Your grandmother might have used a frog to discover she was pregnant with your mother. A laboratory technician injected urine into an African clawed frog and waited. If eggs appeared, congratulations. For 3 decades, this bizarre ritual was medical standard practice worldwide. Millions of frogs traveled from Africa to laboratories across every continent. They revolutionized reproductive medicine. Then, everything went catastrophically wrong. When scientists no longer needed these frogs, they released them into rivers and ponds. The frogs carried something invisible on their skin, a microscopic killer that no one knew existed. Within decades, this fungus had spread globally, driving hundreds of amphibian species toward extinction. This is the story of how medical progress accidentally unleashed one of Earth's deadliest pandemics, and it's still spreading today. The pregnancy revolution. Picture a medical laboratory in 1937. A woman sits anxiously in a sterile room. She suspects she might be pregnant, but has no way to know for certain. Doctors possess no reliable tests. Missing a menstrual period isn't definitive. Physical examinations can't detect early pregnancy. Then, a South African zoologist named Lancelot Hogben made a remarkable discovery. He was studying African clawed frogs for completely unrelated research. These peculiar amphibians lived in murky ponds and rivers throughout sub-Saharan Africa. They were entirely aquatic, never venturing onto land. Hogben noticed something extraordinary about female frogs exposed to human urine. When he injected urine from pregnant women into these frogs, they began laying eggs. Urine from non-pregnant women produced no response. The difference was unmistakable and completely reliable. The mechanism behind this reaction involves human chorionic gonadotropin, or HCG. This hormone appears in pregnant women's urine shortly after conception. The frog's reproductive system responds to HCG by triggering ovulation. Within hours of injection, eggs appear. News of Hogben's discovery spread rapidly through the medical community. Here was a pregnancy test that was simple, affordable, and remarkably accurate. Laboratories worldwide wanted African clawed frogs immediately. By the early 1940s, the Hogben test was standard practice. The process became routine. A woman provided a urine sample. Technicians injected the sample into a female African clawed frog. The frog went into a special container for observation. Within 12 to 24 hours, results were conclusive. Photographs from this era show technicians in white coats holding frogs and syringes. The images seem surreal to modern eyes, but this was cutting-edge medical technology. For the first time in human history, women could know definitively within a day whether they were pregnant. Demand for African clawed frogs skyrocketed. Suppliers in South Africa trapped thousands of frogs from wild populations. They shipped these amphibians to Europe, North America, South America, Asia, and Australia. No continent except Antarctica was left without African clawed frog colonies. The frogs thrived in captivity. They were hardy, easy to maintain, and could be used repeatedly. Laboratories kept large breeding populations. By the 1950s, hundreds of thousands of these frogs lived in medical facilities worldwide. They had become an essential tool of modern medicine. The obsolescence. For 3 decades, African clawed frogs remained the gold standard for pregnancy testing. Then, in 1968, everything changed. Scientists developed the first immunological pregnancy tests. These tests detected HCG directly without requiring live animals. The new tests were faster, cheaper, and more convenient. Results appeared within minutes instead of hours. No specialized animal husbandry was necessary. Laboratories could store test kits on shelves indefinitely. The frog test became obsolete almost overnight. By the early 1970s, at-home pregnancy tests appeared in pharmacies. Women could now test themselves privately without visiting doctors or laboratories. The medical revolution that African clawed frogs had enabled was complete. Their services were no longer needed. Laboratories worldwide suddenly found themselves with massive frog populations and no use for them. These weren't native species that could simply be returned to local ecosystems. They were African amphibians thousands of miles from home. What should be done with them? Some facilities maintained their frog colonies for research purposes. African clawed frogs had become valuable model organisms for developmental biology. Scientists studied them to understand embryology, genetics, and cell biology. These research populations remain in laboratories today. But many facilities saw the frogs as surplus inventory, taking up valuable space. Maintaining aquatic animals requires resources: tanks, water systems, food, staff time. With no medical purpose, the frogs became an unwanted burden. Administrators made a decision that seemed harmless at the time. They released the frogs into local waterways, into rivers, ponds, lakes, and wetlands, across North America, South America, Europe, and Asia. Thousands upon thousands of African clawed frogs entered non-native ecosystems. They began establishing wild populations far from Africa. The frogs adapted remarkably well to their new homes. They were generalist predators that consumed almost anything: insects, small fish, other amphibians, even carrion. They could tolerate a wide range of temperatures and water conditions. They reproduced prolifically. Within years, invasive populations had established themselves across multiple continents: California, Florida, Chile, France, Italy, Indonesia, Japan. Anywhere with suitable aquatic habitat and moderate climate. The frogs spread through waterways, moving downstream and colonizing new territories. They became one of the world's most widespread invasive amphibians. The invisible killer. African clawed frogs weren't traveling alone. On their skin lived microscopic passengers, a fungus called Batrachochytrium dendrobatidis, or Chytrid fungus for short. This pathogen had evolved alongside African clawed frogs for millions of years. The frogs had developed complete immunity. Chytrid fungus lived on their skin without causing disease. The relationship was commensal, neither harmful nor beneficial. The fungus existed. The frogs remained healthy. This balance had persisted for evolutionary time. But Chytrid fungus wasn't limited to African clawed frogs. It could infect virtually any amphibian species, and most amphibians had never encountered this pathogen before. They possessed no evolutionary defenses, no immunity, no resistance mechanisms. They were completely vulnerable. When Chytrid fungus infects susceptible amphibians, it colonizes the skin. The fungus feeds on keratin, a structural protein. As it grows, it produces zoospores that spread across the skin's surface. Infection becomes systemic. Amphibians breathe partially through their skin. They absorb water and electrolytes through specialized skin cells. They regulate body temperature through cutaneous circulation. Healthy skin is essential for survival. Chytrid fungus destroys this critical organ. Infected skin thickens and becomes dysfunctional. Gas exchange decreases. Water balance fails. Electrolytes accumulate to toxic levels. The amphibian's heart rhythm becomes irregular. Eventually, cardiac arrest occurs. Death follows within weeks of initial infection. Most devastating is Chytrid fungus's ability to spread. Zoospores can survive in water, moving between hosts. They can persist in moist soil. They can travel on birds' feet, fishing equipment, hikers' boots. Any pathway that connects water bodies becomes a transmission route. When released African clawed frogs entered wild ecosystems, they brought Chytrid fungus with them. The pathogen found populations of native frogs and salamanders with zero resistance, perfect hosts for rapid spread. The pandemic began. The collapse. The first mysterious amphibian die-offs occurred in the late 1970s. Researchers in Australia discovered dead frogs in mountain streams. The animals showed no obvious injuries or signs of predation. They simply died in large numbers. Similar reports emerged from Central America. Costa Rica's cloud forests, home to spectacular frog diversity, experienced catastrophic declines. Species that had been abundant for decades suddenly vanished. Within a single breeding season, populations collapsed by 90% or more. Scientists initially struggled to identify the cause. No bacterial or viral pathogens could be detected.
Water quality tests showed nothing unusual. Habitat destruction couldn't explain the speed and scale. Something else was killing frogs, but researchers couldn't determine what. Then, in 1998, a team of scientists identified Chytrid fungus on dying amphibians. The discovery was shocking. No one had suspected a fungal disease could cause such devastating mortality. Further research revealed the pathogen's global distribution. Chytrid fungus had spread to every continent except Antarctica. It infected amphibian populations in pristine wilderness areas far from human development. National parks and protected reserves saw collapses. Even species living in supposedly safe habitats were dying. The scale of destruction became clear as scientists surveyed populations worldwide. In Panama's rainforests, 70 species of harlequin frogs declined catastrophically within a decade. Many went completely extinct. These colorful amphibians had survived millions of years of environmental change. Chytrid fungus eliminated them in a generation. Australia lost numerous endemic species found nowhere else on Earth. Gastric brooding frogs, which raised tadpoles in their stomachs, disappeared before scientists could study them adequately. Sharp-snouted day frogs, green and golden bell frogs, countless others declined toward extinction. North America experienced its own amphibian apocalypse. Mountain yellow-legged frogs vanished from Sierra Nevada lakes. Wyoming toads nearly went extinct. Boreal chorus frogs declined across Canada. The pattern repeated across ecosystems. Healthy populations, then infection, then collapse. By 2010, researchers had compiled devastating statistics. Approximately 120 amphibian species had lost 90% of their populations. 90 species had gone completely extinct. Roughly 30% of all amphibian species showed evidence of chytrid infection. This qualified as a mass extinction event, not the gradual decline over millennia typical of background extinction, not the slow habitat loss from human development. This was rapid, catastrophic, pandemic-driven extinction, and it traced directly back to those released laboratory frogs.
The investigation. Scientists began investigating Chytrid fungus's origins. Where did this pathogen come from? How had it spread so rapidly? Genetic analysis provided disturbing answers. The fungus existed in multiple strains distributed globally. But one strain appeared repeatedly in die-off zones across different continents. Researchers traced this pandemic strain's genetic signature. It matched Chytrid fungus samples from African clawed frogs in museum collections. Specimens preserved since the 1930s tested positive. This evidence pointed to a troubling conclusion. African clawed frogs had been carrying the pandemic strain for decades. Every frog shipped to laboratories worldwide between 1930 and 1970 was a potential disease vector.
millions of infected frogs. When those frogs were released into non-native environments in the 1970s and 80s, they brought the pathogen with them. It spread from released populations into wild amphibian communities. From there, it moved through connected waterways and across landscapes. Humans inadvertently accelerated the spread. Hikers walking through contaminated streams carried zoospores on their boots. Fishing equipment moved the fungus between lakes. Pet trade and amphibians transported infected animals across borders. Every vector of human activity became a disease transmission pathway. Researchers also discovered that environmental changes made amphibians more susceptible. Climate change altered temperature and rainfall patterns. Stressed populations had weakened immune systems. They became easier targets for infection. Chytrid fungus exploited these vulnerabilities. Some amphibian species showed surprising resistance. Bullfrogs in North America carried the fungus without dying. They became reservoir hosts, maintaining infection in ecosystems. This allowed the pathogen to persist even after susceptible species disappeared. Conservation efforts began ramping up in response to the crisis. Scientists established captive breeding programs for critically endangered species. They removed frogs from infected habitats before populations collapsed. These arcs preserved genetic diversity for potential future reintroduction. Researchers explored treatment options. Antifungal medications could cure infected individuals in captivity, but treating wild populations was impractical. You can't medicate every frog in a forest. The scale was too large. Some scientists investigated biological controls. Could beneficial bacteria compete with Chytrid fungus on amphibian skin? Early trials showed promise. Certain bacterial strains reduced infection severity, but introducing new microbes into ecosystems raised additional concerns. The fight continues. Today, Chytrid fungus remains a global threat. The pandemic hasn't ended. It continues spreading to previously uninfected regions. Amphibian populations face ongoing pressure. New outbreaks occur regularly as the pathogen reaches isolated areas. But conservation efforts are making a difference. Scientists have successfully bred endangered species in captivity. Some have been reintroduced to wild habitats after local chytrid populations declined. These reintroductions don't always succeed, but some populations are rebuilding. Researchers in Ecuador have been searching for species thought extinct. Some amphibians might have survived in remote refuges. Finding these survivors could provide crucial genetic diversity for recovery programs. Every rediscovered population offers hope. New technologies offer additional tools. Environmental DNA sampling can detect amphibian presence without capturing individuals. This allows monitoring of rare species with minimal disturbance. Genetic techniques might eventually produce fungus-resistant populations through selective breeding. Some amphibian populations are evolving resistance naturally. In areas with long-term chytrid presence, surviving frogs show genetic changes related to immune function. Natural selection is favoring resistance genes. Evolution is fighting back. But the damage already done is irreversible. Those 90 extinct species cannot be resurrected. The lost genetic diversity, the broken ecological relationships, the vanished evolutionary potential, all gone forever. We live in a world diminished by their absence. A pregnancy test seemed harmless. Medical progress appeared purely beneficial. No one imagined that laboratory frogs could trigger a mass extinction. But unintended consequences often emerge from our best intentions. The Chytrid fungus pandemic teaches us that every action in our interconnected world creates ripples. Some ripples become tsunamis. Today, scientists fight to save what remains. They work to restore what was lost. The battle continues, and the outcome remains uncertain.